a ride and look at the flowers blooming and enjoying the spring. Okay, so yeah, so I kind of, as Roy said, I was trained as a statistical mechanics, a theorist, and then I later on I drifted, and at some point, so I got into biological physics, looking at some calculations, problems involving molecular interaction and such. And gradually, I found my pathway into wet lab, okay, and Tom Coleman was the first student, a brave soul that was with the joining a theorist to start a lab. So really, Tom started my wet lab, and we should have even called mine. And so then we learned a lot, so now I run a group that's really basically with mostly with theorists from physics that got interested in biology and do some kind of a simple biology experiments, okay? And then interpret the result and do new experiments and such. I don't think the current state of development in biological physics is really at the stage where you really need to separate between say theory and experiment, because you can do a lot with a simple theory and a simple experiments, as I hope to show you. Okay, so as a kind of an introduction, let me kind of take a historical perspective of sort of how new advances in science came about, right? So we are at the stage where even though if you're not in biology, you hear about in news about, well, how 21st century is the century of biology, we have a biology revolution, and all that. And so I'd like to take a perspective and see when, what does it mean to have a scientific revolution, right? So if you go back to 200 years ago, in the early 1800s, right? So middle of 1800s, what's your image? So I would think about chimneys with the pumping of smokes, maybe steam engines, steamboats, primitive assembly line sort of things, right? So that's the Industrial Revolution, and it is growing through the past century. If you ask what was it behind the Industrial Revolution, right? So I would say, really, what's underlying this is mankind has figured out one trick. Right? That's how to convert heat to mechanical work. Right? And then, so from that, a lot of things came about. And so there you can talk about two aspects: technology and the scientific foundations. Right? So you certainly need technology. You need the steam engine. You need steel, and there's a foundation, the thermodynamics. So, okay, so let's advance for 100 years to the early 1900s. And then we see that throughout lifetime, the development of for the lack of better word, the electronic revolution, right? Starting with transistor integrated circuits, continuing through the 21st century. And if you ask so there's certainly exponential growth, right, in this stuff. And if you ask what's underlying that, well, I would say that, again, we have figured out a trick, right? Well, a trick of nature, that we figure out how to describe electrons and how to manipulate electrons to an extent. Right? Maybe not really the quantum aspect, okay, but that could push us to the next revolution, but certainly we know enough how to manipulate electrons to a certain extent to bring about all of that. And again, let's say there's technology, maybe materials, how to process material, make integrated circuit and such, right? And the scientific foundation would be quantum mechanics. Okay, so now we're in the 21st century, right? And there's all the promise of a wonderful things the biological revolution is going to give us: personalized medicine, designer bacteria to clean up the environment, such and such. Okay, long list of these applications. I do not know, in order to do this, could this be done just by kind of incrementally improving things, which is pretty much what's happening in biology. In biology, one learns about tricks the biological system does, and kind of basically in an empirical way trying to improve things. Okay, but I do not know whether there will be some kind of fundamental lessons about nature that will come about in order to make this thing into reality. I certainly hope so. Okay, but regardless, we'll say in order to reach these goals, one will need to have a accurate quantitative description of living systems, right? And we need to be able to design and manipulate them. And okay, so I leave this blank, but just like in early 200 years ago, in the early 1800s, well, this word thermodynamics means nothing, right? I'm not sure, like, whatever lessons we're going to learn about biological system, maybe this word hasn't even come about yet, okay? But there's a hope that maybe there's something fundamental that we'll learn about these systems. So you can ask for, should there be a new lesson? Okay, so fundamentally new concepts do not just come about every day, right? So these are like once a century kind of a discovery or encountering of a new science. But in hindsight, you would say, yes, maybe one should not be surprised that there's something new here. And the key thing is here, we're talking about very small scale. There's no reason a priori why our intuition about what happens to tennis ball should be applied to the atomic electronic scale, right? So, but that only we learn after the fact. Maybe we'll say, yeah, there's no reason why it should not be that. In the development of thermodynamics, of course, we know a large number is ultimately the key for giving us a new phenomena. Then, so we can ask, is there something here, right, that could potentially lead us to sort of new concepts? And I would say yes, okay, and the main thing is the heterogeneity. So heterogeneity not in the sense of randomness, okay? I come from this discipline of a spin glass, okay? So you can have random interaction and you can look at the statistical averages, the properties and such, right? That's heterogeneous, but then it's kind of with a prescribed distribution. But here, for biological system, a cell is made of many, many components, and every component seems to be unique and special, right? And at least if I were to look up the repertoire of physics, I'm not sure there's anything I can look up that'll help me to deal with something that's specific and very, very different, right? It's something involving thousands, maybe millions of objects that are all different. Okay, and it's sort of come together to do something special. Okay, so just keep this in mind. So I'm going to tell you about this heterogeneity, this biological heterogeneity, not randomness, right? Biological heterogeneity, in a context of a bacterial growth. Okay, so here's a E. coli cell that we grow in our lab. And so if you put it in, so this is a growth curve. The y-axis is the density of bacterial cells, and this is time. And if you put it in some medium with some sugar, some ammonia source, then it can nicely grow exponentially, eventually saturated. Right? So there's this phase where it's of exponential growth, where this lambda used as a growth rate. Now, this growth rate, which is a very good characteristic, it's like taking a pulse, something, right, of this culture. It is dependent on all kinds of things: temperature, pH of the medium, osmolarity, whether you apply drug to it, different type of nutrients. Okay? It's reflected in one number. And also, there are a lot of internal dynamics of the cell goes into it. So some cell can grow faster than the other, given the same situations, right? And that has to do with lots of enzymes, how they are coordinated, how they work together, regulation of the enzyme functions. So let me dive into this a bit. Okay, so what does it take for a cell to become two cells? Well, if you open up the cell, you see it's made of DNA, proteins, and all kinds of things. Right? And just very roughly, for bacteria, and this is also true for pretty much for any kind of cells, 2/3 of the cell is water, cytoplasmic. Inside the cytoplasm, okay, it's water, okay? And about 1/3 is a biomass. If you look into biomass, about 2/3 is proteins, 1/3 is RNA. This changes with the condition, but roughly, okay? Mostly it's a protein and RNA. Okay. And the DNA, I should pay a lot of attention to, is only a very small part. It's the memory, it's the hardcore memory, right? So only a small amount in terms of a biomass. So let me dive into, follow the protein and RNA. Right? So for the cell to double, it needs to make twice as much protein and twice as much RNA, and divide and do other things. At least you need to make all copies of these things. So proteins, well, they're polymers of 20 specific amino acids, stringed together in different orders. How are proteins made? Proteins are made by an object called ribosomes, okay? And so they're coded by a piece of a molecule, the mRNA, and the ribosome basically go on, read on the mRNA and start to put together amino acid in the order instructed by the mRNA, make into proteins. So this is basically, you have to do this, right, for a cell to become two cells, because you have to make most of the, twice as many proteins, which is most of the cell mass. So now I'm on track to tell you about the heterogeneity, complexity in the cell, biological complexity, okay? So for the next 5-10 minutes, I'm going to give you a dose of just a lot of things flying around, okay? And if you feel overwhelmed, that's okay, because that's my intention, okay? But then you do not need to follow the details, right? But that just, in order for this to happen, a lot of the things have to be coordinated. That's my main message, okay? But let me just so that you get a taste of what's actually going on, I just want to walk you through a little bit, okay? And then after that, we're going to kind of simplify, okay? So this is a ribosome. It consists of 50 proteins, and so there are several pieces. The ribosome itself involves RNA and a protein, and most of the RNA in the cells is actually the RNA used in the ribosome, called ribosomal RNA. And about 1/3 of all the proteins in the cell mix is in the form of ribosomes. Okay, so altogether, ribosome takes up about, for fast-growing cell, about half of this biomass is just ribosomes. All right, now, again, another player is tRNA. That is, you have a transcript that constructs that contains a message of what protein should be made, what order of amino acid, and this is done by a transfer RNA, which reads the message RNA, this is the triplet codons, and on the other side attached with the amino acid. Okay, so it brings the amino acid together with the instruction. Okay, so this is maybe a kind of more three-dimensional model of a tRNA. So there's a part that contact with mRNA and the amino acid. So how does this machine work? In the cartoon, but I'm only telling you in the cartoon level, a lot is known about it, and we have some experts sitting in the audience here. But even the cartoon level, you'll see, is quite complicated. Okay, so a ribosome covers 3 triplet codons. Okay, and let's say at a certain stage, so it's reading the mRNA halfway through, and there's already a peptide chain the amino acid chain that's being made. So there is a position, so a position that's where accept the latest codon to come in, and that's open at this point. And so then it's looking for a charged tRNA, that's the amino acid associated with the tRNA, and this thing will go in, and it will sit into this open position, and then the connection will be made, and this thing will move forward, okay, and then it cycles back. So that's how it takes to make a protein polymer. Now that's just the start of it. So then you can ask, well, where does this thing come from? Well, it comes from a soup inside a cell of a tRNA by itself and amino acid by itself. Okay, so amino acids made by metabolism, and the tRNAs, the ones that's used, tRNA that gets recycled back into the pool. So somebody needs to put them together. So there's some enzyme that puts the right amino acid on the right tRNA, okay, to do this. It's called tRNA charging. charge the tRNA with the right amino acid. And then if you were to write down a, say, equation for this charging, you can write down, no problem. So it's chemistry. Basically, there's a flux for this to happen, and depends on the concentration of the enzyme that's doing this, depends on the concentration of amino acid, depends on the concentration of tRNA. Now, what I'm getting at is that there are a lot of variables here. 20 amino acid, there are 60-some tRNAs, okay? And there are another 20 of these enzymes. That's already 100 variables. Each one of these, there's several parameters, okay? So for this limited system, just for the one step of protein synthesis, very minimally you have something of the order of 500 parameters. It's working. But that's just the beginning, okay? To make amino, so where do amino acids come from? If I'm feeding a cell with a glucose, then there's a way the cell basically does chemical modification and converts glucose into amino acid. So here is an example of what is done. So a bunch of enzymes modify the molecules and eventually convert into the amino acid. Okay, so there are a number of enzymes. Each one has a number of parameters, and there are 20 such things, and actually many more, okay, to make it other stuff. So now you put it together, so there's at least 500 enzymes, more like 1,000 enzymes to make these 20 amino acids. Each enzyme has parameters that describe how it works. And then the intermediate metabolites that's in between, the intermediate step between glucose and this amino acid. Okay, so altogether, you're talking about many thousands of parameters. But that's still just the beginning. Okay? Well, cell use lots of regulatory strategy to make sure these things are coordinated, how many enzymes to make, how fast they should be going, this and that, okay, traffic control. More proteins do this, more parameters. And all of this have to work not only in one condition, they have to, so this already several tens of thousands of parameters. They have to work under different pH, different temperatures, and all of that, right? So every one of these parameters, if you imagine to figure out how they depend on all of these things, you have really explosion of parameters. Okay? So minimally, to get this, what's behind this exponential growth is really, millions is really an underestimate of the parameters. Right? And we also have protein-protein interactions, and it gets combinatorial. I'm not even keeping that. Okay? And that's just for simple bacterial cells. Now, what about something more complicated? Right? So before the end of the day, I mean, we are talking about really macroscopic number of parameters. Okay? So maybe then, then if we have basically we have a system that has really so many parameters, you can weight the parameters by their weight. Okay? So that's a kind of problem we're dealing with in biology. Okay, so that's kind of supposed to, I guess, wake you up or make you depressed or something, right? That's a challenge. But on the other hand, so this is a physics department, so we have some experience dealing with problem with modes of parameters. Right? We have physics students here? Physics students? Right. So tell me a experience you've had of working with modes of parameters. Everybody has worked with it. Anybody? Modes of parameters. Okay, you've all taken thermodynamics, statistical mechanics, right? So if I ask you to look at a property of a gas in this room, okay? And I tell you that gas is made of molecules that roughly obey Newton's law, right? Then you can start by writing Newton equations. And each Newton equation, for each molecule, you will need initial conditions, right? 6 variables. And if you have Avogadro numbers, you're talking about many modes of parameters, right? So, of course, we don't do things that way. Right? And so instead, there are relations like this that has a single parameter, which is the gas constant R. So This is what I mean by the example of a dimensional reduction. That if you look at the problem one way, you have many, many, many parameters. It's a hopelessly complicated problem, right? And but then, at least if you look at certain properties, things may be much simpler. So where does this dimensional reduction come from? Okay. Then you can, okay, so then immediately you can try, well, can I then go from here to there? Okay. Well, not very easily, right? Boltzmann tried, and he committed suicide. Okay, so fortunately, right, when people were looking at thermodynamics, they didn't know gas were made of molecules, okay? And so there was all this phenomenological description of thermodynamics that came before Boltzmann, right? So it's only, so these would develop two separate branches of science, right? And then with the benefit of both, then Boltzmann comes along and connected them together. So there's statistical mechanics, and ultimately at the kind of a utilitarian level, what is statistical mechanics? Statistical mechanics is a shortcut, right? It's a shortcut that, so in principle, so we're interested in the density function of the system. So all the general coordinates Q and P, right? So there's some, this eventually described by some density functional that depends on all of the P's and Q's of the coordinates of the problem, right? And ultimately, we're saying that in the long time limit, this is actually given just by a function of these, all these 10 to the moles of a parameters of coordinate variables through a single function. That's the shortcut, right? We don't have to follow the dynamics. We just need to know because we just know that the dependence through the Hamiltonian. Where does that come about? Okay, we can talk about, in this particular case, where statistical mechanics came from Hamiltonian dynamics, conserved quantities, and that sort of things, okay? But ultimately, this is the effect that because you have this Hamiltonian, right? And you have this reduced description. So eventually, it's a simple description because all the variables collapse into one variable. So can we do something here, right? So what we're interested in is to figure out the whole. So what plays the role of thermodynamics in biology is physiology. What is the whole doing, the behavior of the whole, okay? And what plays the role of Newtonian mechanics is molecular cellular biology, what the molecules, interaction of the molecules, interaction of the cells, right? So can one go from here to there? Well, because this is certainly too hard at this stage. So what I'm going to be talking about is for bacterial cell, for the problem of a bacterial cell growth. Still, there's an issue. Can we go from molecules to the behavior of a cell? Okay? And so this is the kind of thing on the molecular level that I mentioned. And this is the kind of thing at the physiology level. Okay, so growth, okay, which involves the entire cell, not just a few molecules. It turns out that in biology, very little attention has been, in the last 50 years, very little attention has been paid on this side. Most of the effort has been here. So then when we went in to look at this problem, we actually decided to spend much of our effort on this side, to establish the thermodynamics. Okay, and so I'll show you an example to see what I mean. The simple relations describing behavior of the whole. And then so that's where we spend most of the time on this, what we'll tell you about. And then from the knowledge of the two, then we can put something together to connect the two, okay? And the trick of a dimensional reduction, well, actually we encounter something very similar to what we're having in statistical mechanics. That is, in principle, okay, so there turned out to be an important object, a regulatory function that determines how fast cell growth. This is called chi-R. And it depends on many of the molecules inside the cell, if you just want to write down microscopics, okay? But then it turns out that this is given, actually the way that depends on this molecule is through a single function, mostly through something, okay? And as a result, you have a similar type of a structure, and basically that allows you to have simple relation at the physiological level. Okay, so that's kind of the kind of the overview of what I'll talk about. So the result of this connection, just to give you, in case time run out, what kind of thing can we do with it? So here's the example. We're growing cells. I show you the exponential growth curve. But here, we do a switch on them. Okay, so initially, cells, so just focus on the black curve, the black points, they're just the density of cells. Okay, so normally they would just be growing exponentially. At some point, we make a switch of a medium so that here there's a medium they're happy in, nutrient that they're very happy in, they're growing fast, and then they switch to something that they don't like as much, they're growing more slowly. And there's a transition. Okay? So by knowing this kind of a connection, then we were able to produce a simple theory that describes this entire transition without any parameters. All we need is the initial growth rate and the final growth rate. Okay? More than that, we can predict what's going on inside the cell, what the ribosome is doing, what the proteins are doing, and so forth. And the same theory, if we just switch the initial and the final conditions, so we go from here's going from fast to slow growth. If we switch from slow growth to fast growth, and the things are inverted, but then the same theory can capture all of the behavior. Okay, so that's the kind of the thing you can get at the end. Okay, but then I want to spend time telling you about the physiological characterization, the thermodynamics, the macroscopic relations. Okay, so this starts with a set of bacterial growth laws that's discovered really starting from the beginning of late 50s, going on in the 60s and 70s. So this man here, Ole Moller, he's a Danish microbiologist, and the kind of things he did was known as the Copenhagen School of Biology. In their lab, in the 50s, they grew cells in different type of nutrients, so they get a range of different growth rates. So each point here is exponentially growing culture. Okay, so there's a x-axis is the growth rate, and the y-axis is the ratio of the total RNA to total protein. Okay, so they have exponentially growing cells. They take a sample, they measure the total RNA contained in it, they measure the total protein contained in it, take a ratio, and hey, there's a straight line. Okay, and these different type of growth rates are arranged by limiting nutrient. Okay. All right, so what does the RNA protein ratio mean? Okay, so I told you most of the RNA is in the form of ribosome, and the protein is basically most of the cell mass, and the water mass ratio is constant, so roughly per total protein is like per water volume, per volume, and this is like the concentration. RNA protein ratio is roughly the concentration of ribosomes. And ribosomes are involved in protein synthesis. All right, so how does one understand this? So Moller so at that time, people doing biology are rather quantitative, to think about in terms of numbers and the mathematical relations. And so he had a very nice way of thinking about it. So assume all of the ribosomes are efficiently working to produce proteins. That's what they do. And all of the protein that's produced are stable, and as the cell grows, they just kind of get diluted. Right? So then the rate of a protein mass accumulation is just the rate of how fast ribosome work. Ribosome elongates. And so then for exponentially growing cells, then the total protein mass is just growth rate times the protein mass. And how fast ribosome works is just the number of ribosome measured here in the mass of ribosome multiplied by how fast they work. Okay, so then if you equate these two together, then you see indeed the ratio of the ribosomal mass to the total mass, we call this phi r, fraction of the protein that's ribosome, should be a linear function of growth rate. Okay, with the slope being 1 over the elongation. All right. So, and so this has been tested in many different ways, and the Moller's interpretation mostly correct. And so we can think about the conclusion is that this linear relation means that most of the ribosomes are engaged in translation. All right. So this result stood for about 50 years. Okay, and not too much sort of happened after that. So we were searching for something, and at the time, Matt Scott, my postdoc, and I, we encountered this result, and we got very interested in it. That biology can have something so simple that the result doesn't depend on the specific stuff you're feeding it, just kind of as a general relation. Okay, but then we felt that it is missing a partner. Okay, so in this picture, the two things that determines how fast cell growth. One is the type of a nutrient that's being fed into the cell. That's what generates the x-axis. And the other is how fast ribosome grows. Okay, so the two things that's important for cell growth, one is how fast ribosome work, and the other is what kind of nutrients come in. And only one of them is being varied. Okay, so it felt to us it's like it's an incomplete picture, and so we set about to look for what in physics language we call a orthogonal perturbation. Like a phase diagram of a magnetic system involves both the magnetic field and temperature. Right? And if you have only one of them, kind of it's not a closed system. So we're looking for an orthogonal kind of a perturbation. And so what we did was we said, okay, let's fix the nutrient and vary this how fast ribosome work. And this could be done by, say, feeding these bacteria with some drug that slow down their ribosomes. Most antibiotics actually work by blocking ribosomes. Okay? And when we did that, we got a surprise. Okay, so this is a pick one. So one of these points is one set of a nutrient, say glucose, and you feed a different amount of a drug to it, and so cells slow down growth, but then they dial up ribosome. Not only they dial up ribosome, qualitatively it's well known to microbiologists, it's tracing out another straight line. Okay? And we did this for different type of nutrient, and they all went up with the following straight line. I'm not showing you the results. Okay. So wow, there's another straight line. So now we have two straight lines, okay, for two variables. We have a closed system. But we did more than that. We also look at, addition to ribosome, what are other proteins doing? So this is for carbon limitation. We are looking at this protein that's responsible for bringing in carbon, so a transporter. Okay, and we see that again, it's making a straight line. So we give a different amount of the sugars to grow at a different rate. So the more we limit, the slower it grows. But then it's dialing up this transport protein, okay, this uptake protein, in a very measured way, and to follow a straight line. Then we say, well, what if we do also what we did here? So we fix the carbon source, and we inhibit with a drug to slow down translation. Then we get another straight line. Okay, we get so surprised that this behavior, this is at the behavior because measure you're plotting against growth rate, and the cells are just drawing a straight line. Okay, now before this point, we were looking at sort of how protein interact with each other, with each other, with gene regulation, all that. Everything's complicated, right? Each thing is described by several parameter Hill functions. Everything is nonlinear. Okay, but here we have these simple linear relations, and we did a lot more tests, and basically just keep on seeing straight lines. Okay, so then eventually we were able to construct a very simple phenomenological theory to capture all this data. And here's the theory. They consider this pi here represent all of the proteins the cell makes. Okay? And we divide them into several groups. Ribosome, that's responsible for translation. These guys are responsible for bringing carbon, nutrients for growth. And then there's something about biosynthesis. I did not mention, but it's a similar thing to convert the glucose into different amino acids, for example. Okay, so we declare that in order for a cell to grow, okay, faster to grow, the more of this ribosome fraction you need. That's given by empirical data. Faster for a cell to grow, you need to have more protein devoted to uptake of a carbon. That's this straight line. Okay, and similarly for biosynthesis. Okay, and then then we have we know empirically that there's a fraction of the proteins that's basically, regardless how fast the cells are growing, their amount do not change. So they're like the overhead, okay, like the social security system, whatever. Regardless how economy is doing, you just have to pay that, okay? But the others are needed in different amounts depending on how fast the cells are growing. So there is a constraint. The 4 of them have to add up to 1, or the 3 of them that are depending on the growth situation add up to about 50%. Right? So we have 4 equations, 4 linear equations with 4 unknowns that completely define the system. Right? So you can think about this, what system we're talking about, a simple system of resistors in series. Okay? So in this analogy, current, so the current that's flowing through the system will be the growth rate. That's the flux of a metabolic flux that's going through the cells. Voltage across each component will be this phi. That's the enzyme fraction. So to push more flux through, you need to have more enzymes. Okay? Then in this context, these guys will just be like a Ohm's law, linear relation between current and voltage, right? And they add up to a constant. The total voltage is just 1 minus this Q fraction. Okay? So it's a system now with basically very few parameters, if you think about it. So nominally, there's 3 parameters here. These are the conductances of these elements, okay? But these are actually the variables I'm tweaking, right? So they're not even parameters. They're actually my input. So there's really 1 parameter, it's just this Q. That is what fraction of the cell is devoted to non-growth related kind of thing, the overhead. Okay, so it's a theory with a single parameter. All of these basically vary and to generate these relations. Okay, so now we can try to make some predictions. Right? So one prediction we can make is for this picture is now if we change this non-growth fraction, what will happen to cells? Well, this involves many proteins. It's difficult to change directly. But what we can do is we can kind of force a cell to make something that's not useful. Okay, so like to increase this Q fraction. Okay, so theory, this simple picture would just predict that if you force a cell to make something it doesn't need, then that's like increasing this non-growth related fraction, and everything should be squeezed, and the growth should slow down just like that. Eventually, when this fraction of the useless protein approaches 1 minus Q, that growth should stop. Right? That's prediction, 0 parameter prediction. This is the data. Okay, so force a cell to make some protein. Okay, each line is basically for a different medium, so that in the absence of this perturbation, they're growing at a different rate. Okay, so in each of these growth medium, you can force a cell to make some protein it doesn't need, right? And it just kind of goes down. Now, of course, I can adjust this parameter to make a better fit, but it's not a fit, right? It's just a prediction based on numbers determined by other experiments. Okay, now, people were doing a lot of experiments like this in the early 90s, because that's the beginning of recombinant technology, and people want to use bacteria as a cell factory, so you make E. coli to make insulin, okay? And so that obviously slows down cells, right? This is saying the more you make this protein that the cell doesn't need, the more it slows down, right? But it's predicting a particular way to slow down. So then we said, well, our result is for one particular strain we're looking at in our lab, right? And it's for a particular protein that's expressed, so we compare to other people's data. So here's a comparison. The light blue is the same as, that's our data. This is for glucose medium where many people look at it. And so these are the other people's data for different type of protein that they force the cell to express, right? And you see they kind of congruent. No, it is biology, okay, but they're kind of falling together. Okay. All right. So this gives us the first confidence that, hey, maybe at least for a certain aspect of biology, there's something simple. Okay. In some sense, we'll talk about universality, right? That there's a similarity of behavior that did not depend on details. Right? Keep in mind, millions and billions of parameters underlying all of this, right? But it's doing something that doesn't care about all these parameters. All right, so then from this work, then we went through basically, we used this picture to look at many systems, quite a number of these are sort of outstanding puzzles in biology over the last 50, even 100 years, okay? And we were able to resolve quite a number of these puzzles. Okay, so this is a list of papers that's an application of this picture. I'll just tell you one of them very quickly. Okay, so there's a puzzle called the Warburg Effect, or basic wasteful metabolism, right? Cells have two ways to generate energy. Okay, so you can take a glucose and burn it off completely into CO2 by what's called a Krebs cycle, and it generates many ATPs per glucose. ATP is the unit of energy currency. Usually you expect this to happen when oxygen is present. It's called the respiration. Okay, in absence of oxygen, then you cannot burn things, and then cell can ferment. Okay, that is, it uses this kind of pathway halfway, and it spit out acids. Okay, we cannot completely burn things into CO2. And so this is what the cell has to do under anaerobic condition when there's no oxygen. But it turns out for many cells, even you give it oxygen, it still does this. So it's called a wasteful metabolism. Okay, it's been a puzzle for a long time. Why do cells do not burn carbon, use carbon efficiently, and just kind of spit things out halfway? All right, I show this case here for bacteria, but this is actually first discovered in the context of a cancer, that the cancer cells, they take glucose and then spit out lactate. Okay, they don't use nutrient efficiently in that sense. Okay, so you can think of many possible factors, but then we did one experiment just to look at how they are excreting this this acid, in our case, acetate, as a function of how fast cells are growing. And we see a straight line when plotted against growth rate. Okay, so fast-growing cells excrete a lot, and then a straight line, and then there's a threshold linear behavior. When we want to see this, then so okay, maybe there's a simple reason behind this that's not specific to the many reasons that has been mentioned. And so we went ahead and build a simple model that taking into account the proteome that's needed to push for fermentation and respiration, account for the mass, and this quantitatively explain this kind of effect, and also predict some novelties that were validated by experiments. Okay, so basically the gist of the story there is that you need autoribosome. The autoribosome are busy making things. Okay, so it turns out that when the mediums are good so that they can grow fast, then they give up making this pathway that will consume a lot of ribosome to make. So we have lots of resources. You don't need to save the resource. You want to grow faster. So you direct your ribosome to make the lean proteins that can allow you to grow faster. All right, I'm going to skip this example, no time. But basically, I guess one thing to say is that there are more straight lines. So this is the osmolality stress, okay? And again, very simple relation between perturbations and how fast cells grow. Just straight lines everywhere. So what is behind this? The cell obviously somehow can measure how fast, it knows about how fast it's growing, and it's regulating things, right, in a simple way. So what's the magic? How does the cell make these straight lines? So let me give you a cartoon picture here. That is, so you have a nutrient, external nutrient, that gets pumped into the cells and converted into some central metabolized amino acid, ATP and such. Then they're then assimilated into protein by ribosomes, right? So that's kind of the standard sort of bird's eye view of what the cell is doing. That's the cellular metabolism, right? And the ribosome make all these different proteins. Among these proteins are these uptake proteins that's responsible for converting external nutrient into the internal metabolites. And they're the ribosomes that's responsible for converting amino acid into proteins. a central variable here is how fast ribosome works. So we call this, it's an effective translation speed of a ribosome, sigma. Okay, pay attention to that. So as I said, of the protein that's the ribosome make, some are for uptake proteins that's needed to bring nutrient, and some are ribosome themselves, right, to sustain the exponential growth. In fact, ribosome making ribosome, that's a catalytic loop, and it turns out to be the sort of central parameters. So this is the sort of a catalytic loop that determines how fast cell grows. This is positive feedback, and so this chi r, chi is a fraction of the total protein up that's devoted to a particular protein. So chi r is a fraction devoted to ribosome. Okay, so that is the key parameter that controls the degree of a self-catalysis. Okay, so this is about regulation of ribosome. How much of the protein that's made gets to be ribosomes. Now, it is known through many years of biochemical studies that this central quantity, how much ribosome makes ribosome, is controlled by a strange molecule called PPG, guanine tetraphosphate. Okay, it's a modified version of this nucleotide G. And that itself is somehow inhibited by the neutron status. So the final result is that if you have a more amino acid, then it orders more ribosome to be made to consume the amino acid. Okay, qualitatively it makes a lot of sense. Right? You measure the substrate of it. So your enzyme, to know how much to make this enzyme, you look at your substrate. If you have lots of substrate, then I make more of this enzyme to consume the substrate. If you have a little bit of a substrate, then I reduce the enzyme production. Okay, qualitatively, that's understood how the system works. Okay, more specifically, this is done by a protein called RAIL-A, and it senses this uncharged tRNA. So when the metabolite levels are low, amino acid level low, tRNA gets less charged, and that's what basically sets off PBGPP. So qualitatively, it's fine at this level, and this biologists understand quite well, and it explains roughly the balance. But if you think about it quantitatively, this is a nightmare, okay? Because you have a central quantity you want to control, how many ribosome makes ribosome, how many ribosomes you should be making. But it depends on all of the different amino acids. You need to listen to all 20 amino acids, not only that, all 60 tRNAs. Okay? Because it could be any one of them is missing that you cannot translate. All right? So this is a problem where the central control needs to kind of survey all of these different variables. Right? So just bring this back in again. Right? All of this mess in there. Somehow you need, but any step could go wrong. Okay? So then one can ask, well, how to figure out such a function? Well, we take the view that maybe this difficulty is not just for us quantitative biologists trying to figure out about the cell. Maybe this is a difficulty for the cell also. How does it listen to what its internal state is, right? You know, if you're a bag of proteins, how do you know how fast you're growing? Right? So what information, out of all of these zillions of molecules, how does the cell know how fast it's growing, or how fast it should be growing? So at this point, we got a break by some information from structural biology. That is, so this is a cryo-EM structure of a ribosome cutting action, and here are two tRNAs. So just in comparison, here's a ribosome, there's 3 positions. Okay, so these are 2 positions of these 2 positions. And this is the 3rd position. They arrange it so that there's an uncharged tRNA that's sitting in there. And what you see is that there's one protein that's sitting behind this structure that's colored in different parts of this protein colored with different colors. That's this magic protein RAIL-A that's responsible for making this signal. Okay, so the message that comes out of the structure is that basically, so RAIL-A is a protein. So this is the part that makes this protein this small molecule PPGPP. Normally, this RAIL-A kind of cuddles up and it's not doing anything. Okay, so this picture indicates that this rarely becomes active when it's in a situation that the tRNA is not uncharged tRNA gets into the ribosome, right? So this A site should be charged tRNA going in to do the work. But if you have lots of uncharged tRNA, the uncharged tRNA also get in. When it gets in, then they can split with the rel A, and then it can send some signal. Okay, so the qualitative picture we get from this is that perhaps this is suggesting that rel A is a way for the cell to monitor the state of elongation. Basically, what fraction of time this gets unoccupied, just kind of sitting there waiting. Okay, so basically then it changes our picture from one where some mysterious interaction out of hundreds of variables that's affecting the production of this signaling molecule into a very simple one that is, signal is actually coming from the state of a translation. This itself is dependent on how fast you translate is dependent on all of these molecules. Okay, so in other words, we have a situation where the dependence of the many molecules is through the dependence on one object. Okay, that's how fast cell translate. To translate, it needs all of these hundreds of these molecules. So at this point, remind you again, this implication one has in cyclical mechanics. It has the same structure. Okay, and this collapse, so this dependence, that the dependence on many molecules through dependence on a single molecule, a single activity. That's all we need to basically to fix the system. Okay, so with this, so if we take as an ansatz with that, the dependence of this key regulation function is through this translation, then one can write down a number of constitutive relation equations that basically describe how ribosomes and these uptake proteins will be synthesized and diluted and such. And then through the definition of this translation speed, one can write down a single ODE to describe how this central control variable will be changing. Okay, and furthermore, so it involves two regulation functions which can be fixed by the steady state properties. So we know everything about steady state through these growth laws. So everything about the system is known. It's one ODE, okay, that just all it needs is the initial condition. Okay, that to tell you what the system we're dealing with. And so this is the solution. This can be solved in closed form, and you can then basically, from that you can that's the prediction that I showed you from the beginning, how cell density changes, how the ribosome changes, and how the uptake protein changes. All just come from solving this equation. All you need is there's only one parameter, that is what state is switching to at time 0. And again, the same equation, if you run backward, produces the other shift from a fast medium to a slow medium. More than that, you can look inside of cell. These days, there's technology to survey proteins, mRNAs, what's happening inside the cell, and the theory describes how inside the cell in blocks what protein groups are changing. All right, so I'm basically done, but let me just summarize. Right, so the key thing we're talking about here is the cell figures out how fast it grows and controls its behavior by monitoring how fast it's translating, making proteins. Okay, so this, here G is a symbol for this concentration of PBGPP. Okay, the state of translation affects this PBGPP concentration, which then through known mechanism regulates how much ribosome to make. And this is the key part. So at the time when we published that paper I just mentioned, this was a hypothesis. Okay, so then recently we actually went in and measured these quantities. And so again, this is a growth shift. This is density of cells. In a fast median, we switch to a slow median. There's some lag, and then goes on again. So we measured the translation. That's this translation speed. This is sigma during this lag period when it's changing. So after the switch, it drops close to 0, and then gradually recovers. And we measured PBGPP. It spikes up and then goes down. Okay. Now, if you make a scatter plot of the two, you get very simple linear relations. Okay, so this is to indicate that indeed the cell, the central signal is listening to the state of translation. Okay, and we can construct a very simple model to describe how this linear relation came about, but I'm out of my time. But this is the simplest kind of a system where synthesis depends on how the fractional time that this A set is unoccupied give you this linear relations. Okay, so we have here a case of activity-based mode of regulation. This is a rather different concept for what's predominant in biology, biochemistry. Most people commonly think about regulation as being kind of so there's two kinds of regulation that people talk about. End product feedback inhibition. So if you're making some stuff and you have lots of it, then you stop the process of making more. Okay, that's sensing the concentration of end product inhibition. It's very well known. Another type mentioned is that you listen to the input. If you have lots of a supply building up, then you make the stuff to eat up the supply. This type of these two strategy, okay, they're commonly used for simple systems. But it's unclear for a complex system, right, where you have to listen to hundreds of variables, what you should be listening to. It's not easy to see how this would work. And in fact, I would say anytime you have a concentration-based regulation, there's a curse because then you have to talk about parameters. When you talk about concentration, there has to be the binding constant, this and that, many parameters. Okay? And what the cell seems to be doing here is it's listening to actually the activity, okay? And then free itself of these parameters. So finally, just to make a kind of an analogy to take home, so if you're running a big factory, right, making complex assembly of the stuff, you're hiring many workers, each doing their own things. How do you know how many workers to hire? Right? So you could, so that is the two people. You could look at the order, how many things are ordered, but then you have to keep track of sort of each worker, how many products it's making and how they are putting together and so forth. It's not so easy if you want to keep track of every individual product. Similarly with the input-driven case, look at the inventories. But then what our study suggests is that in the case of E. coli, it is simply looking at the number of idle workers, right? How often workers are taking break, okay? If you see a lot of people taking break, you fire some workers, okay? And the things will be fine, will be better, okay? And if nobody's taking rest, then you need to hire more workers, okay? Use it or lose it. All right, so I'm done. And so this is a simple picture illustrating the connection between small molecules and the behavior of the cell. And of course, our goal is to push to more complicated organisms, more situations. And we think the key here is that we need to know more about sort of what phenomenologically at the higher level what things are doing, the simple relations, okay, before we can make connection to the molecular parts. So this body of work has been building up over the 10 years by a large number of great students and postdocs involving, through which I learned a great deal. And the particular work on growth transition kinetics was done by David Erickson and Severin Schenk, who is a visiting student from Munich. Okay, so finally, the motto of our lab is a quantitative and predictive understanding of organizing principles of living systems. Thank you. Yeah.
How do you know how many relevant parameters there are?
How many what?
How do you kind of develop an intuition for how many relevant microscopic parameters?
Yeah, how many parameters you need depends on what phenomena you're looking at, right? So here I'm looking at sort of phenomena where just by looking at the result, looking at the plot, you see that there's a hope to describe by a few number of parameters. But if you're asking a question of a particular about which protein interact with which, then of course you need many more parameters. The same with a gas. If you ask detailed question about 5-body correlation, you need to know a lot of parameters.
Do you find like sometimes there's new relations?
The what?
Relations between these a priori independent parameters?
So again, the key here, right, to look for to put yourself in a position where you can benefit from dimensional reduction is you need to ask. Basically, you need to find in the physics world. You need to find the coarse-grained variable, the order parameters, right? How many order parameters, right? So you need to be asking a question that has a chance for a simple answer, okay? And our experience at with bacteria is actually if you ask the questions that involving the whole system, right? What the bacteria cares about, but the behavior, it seems to be simpler than the number of molecules imply, right? I mean, you look at your body movement. I mean, if you think about to go this, what neuron has to fire, what muscle have to pull, and so forth, it's very complicated, right? But then maybe there's another way to think about it so that it's much simpler.
So how universal the formula you find to like for example, you know, in experiment you use certain cells. Let's say to grow other cells, you get the same rules, but there's some parameters.
Right. Being a statistical mechanic, I mean, it's very easy for me to kind of to hope that's the universality, right? But I've been in biology long enough to be very careful about that. So all we claim is that for our system we see this, and that's already, I mean, not easy, okay? Since our work dating back to maybe quite a few years ago on the steady state properties, people have been looking at other systems, right? And in fact, I kind of want to not want to look at other systems because I will have a bias going in, right? You need to let other people look, right? And it seems to seeing other bacteria in yeast. I mean, my colleagues upstairs were looking at the cancer cells. They also follow these simple rules. Okay.
Does this mean there's the same regulation mechanism?
It's not mechanism, but perhaps it's a strategy. Yeah.
So in the end, doesn't this just boil down to there's a very small number of rate-limiting steps?
Yes. So in the case where we can predict the growth kinetics, right? We didn't even need any kinetic parameters. Okay.
Right, but there are processes.
Yeah. Right. So yes. Ultimately, yes. There are a few. There's a few rate-limiting steps, and the ribosome basically. That's what it is. Okay. Now, but then unlike in physics, where we can say, well, but then can ask why is that? Okay. The biology has so many time scales. I mean, how do you know what's the limiting step, right? But then our view is that maybe, an unorganized thing is not just difficult for us to calculate, but it's also difficult for the cell, right? The cell needs to do something in kind of a reproducible way, in a controllable way. And maybe this is just an easy way of organization so that it can get a hold on something. So if there's a system where there is a limiting step and they can control that, and then everything's set.
So something like Linsky's experiments where you evolve bacteria and
Yes.
they speed up.
Yes.
That implies that somehow they're changing this population.
Yes. So Matt Scott actually has been repeating this characterization for Lansky's evolved strings. Okay, and he finds some very interesting things about what. So in the simple model, there are only a few things that could be changing. Okay, I cannot describe his work, but he finds some very interesting thing. It's just a simple change. Yeah.
You were mentioning guanosine tetraphosphate being the signal.
The what?
Guanosine tetraphosphate regulating the speed of ribosome synthesis.
The other way around. So guanine tetraphosphate is known to be the agent that regulates ribosome synthesis, right? But the question is, what is it sensing?
But in the end, there is a strain, or there are strains out there where guanosine tetraphosphate is null. Everything is mutated. There is no guanosine tetraphosphate.
Say that again?
There's
Yeah.
LA can be deleted.
Yes.
And also it's
Yes.
relative, which is necessary.
Double deletion, right?
That signal can be removed.
Right. Okay. Yeah.
What is going
Right.
on there?
Okay. So we try to look at. So those strains are very difficult to work with. Okay. So you have to. So we cannot do our kind of experiments. Right? So basically, those strains you need to feed it with all amino acid. Right. Yeah. You have to be in rich medium, and so there's nothing for us to study, right? Because we're talking about how does it control. Right. So you have to. I need to be able to change.
You
but I cannot change. Huh?
You could still use antibiotics to slow it down.
Antibiotics for these rare, okay, maybe, but so these strains are already, because they mutate like crazy. Okay, yeah, so there's no, so in this case, there's a, so we will say that they're not regulated, right? So they're just kind of set in some rate, okay? And so we try to look at them. This is very difficult to work with.
You can measure just the maximum growth, maximum synthesis rate?
What?
Because you're not down-regulating it with guanosine tetraphosphate.
The maximum protein synthesis rate?
Maximum ribosome synthesis.
Yeah, what about them?
Because you have no guanosine tetraphosphate to shut it down.
Right.
Then assume that you would measure.
Yeah. So, but then the thing is that so for these strains, but you need to complement with the form. They cannot adjust. They cannot adjust speed. Okay, and you kind of you just put it in there, and you kind of pray for that they won't mutate in a little time. Yeah. So we don't know what to say. We very much like to look at those. Okay, but these things pick up mutation like crazy, right? So it's not easy. But maybe you can tell me how to study them. We very much like to study them.